Hello everyone, this lesson is going to be on the cell membrane and transport across the cell membrane. Cell membrane around the outside of the cell, separating the interior from the exterior, also referred to as the plasma membrane. But as we'll see, it does much more than just create a border between the inside and the outside. When I am talking about membranes, just kind of keep in mind as well that there's not just the membrane around the outside, the cell membrane, but around every single one of the internal organelles in a eukaryotic cell, cells that do have internal organelles, there's also a membrane. So we have the nucleus here, and there's a nuclear membrane. This is the mitochondria, and there's a double membrane. Golgi apparatus, endoplasmic reticulum, all of these are also going to have membranes. Each one is going to be a little bit different, but their basic structure is going to be pretty much the same with all of those different membranes. So yes, it does surround every cell, but like I said, it is much more than a border, a wall. It is referred to as selectively permeable. So it's really the membrane that determines what is going to pass from the outside to the inside and what's going to go in the opposite direction as well. So it is going to regulate the movement of materials into and out of the cell. Picture on the right hand side here, this is an electron micrograph, and we're going to talk a fair amount about the actual structure of the cell membrane. And we can see in this picture that there are two lines, an inner line and an outer line, and that's because the membrane, all membranes, they are made up of a bilayer. Bi meaning two, so in fact there are two different layers that are kind of sandwiched together to form a membrane. Nice picture here that is showing the structure, fairly complicated structure. Membranes are incredibly complicated, and again, each one is kind of unique. One cell compared to another will be different. Internal organelles will each be different as well in terms of certain structures that they do have embedded in the cell membrane. So first of all, the bulk of the cell membrane, it is made up of what is called a phospholipid bilayer. So these two different layers, we'll take a closer look at these, but they are phospholipids. And for now, I'll just kind of point out that lipids are a fatty material. And if you think of common fatty materials that we encounter like oil, oil doesn't mix very well with water. So the lipid portion of the bilayer tries to avoid water as much as it can. And keep in mind that the interior of the cell, the cytoplasm, it is mostly water. And the outside of a cell, well, it is mostly water as well. So you will have a portion of the phospholipid bilayer that tries to avoid that water as much as it can, and other portions that are going to come into contact with it. In addition to this phospholipid bilayer, though, there are a whole bunch of other things that are studded or embedded in the phospholipid bilayer, and this creates a mosaic-like pattern. Things stuck in the background bilayer, and what these things are that are stuck in, they include proteins, glycoproteins, lipoproteins, and cholesterol. So let's take a closer look at this picture here. So if I can just put a box around one portion of this membrane right here, this is strictly phospholipids. There are no proteins, there are no sugars. This is strictly the phospholipid bilayer that we have here. If I divide it right through the middle, then we have this region that's closer to the outside of the cell. I'll just put an O for outside, and a region that's closer to the inside of the cell. Everything in yellow here, as we'll see, that's kind of the fatty portion that doesn't like the water, and that's why it's sandwiched in the middle of this bilayer where there is no water. So there is water inside the cell, there is water outside of the cell, but in between there is no water that you find in there. So the arrangement that we do have then of these bilayers is these lightly colored ovals that we see here. Those are the portions of the bilayer that will like water, and the ones that are shown in the yellowish color, those are the portions of the phospholipid that don't like the water. But of course, this membrane is much more complicated than that, and there's a huge amount of complexity outside of the cell, what's referred to as the extracellular matrix. and inside of the cell as well, where they have all of these that they're labeling as microfilaments and cytoskeleton. Dealing with the actual membrane itself, though, we see a whole bunch of other things stuck in, embedded right in, creating this mosaic-like pattern in the phospholipid bilayer. So all of these kind of purplish, lightly purplish colored blobs, they are all proteins. And all of these ones that I'm putting a check mark on here, they all pass directly through from one side to the other side of the phospholipid bilayer. And these ones that pass directly through, they are referred to as transmembrane proteins or integral proteins. And they're going to be really important for our discussion involving the transport of molecules from one side of the membrane to the other. Some of these other ones are restricted to the inside or the outside. So this one here is restricted to the inside, this one restricted to the inside. It kind of looks like probably these ones are stuck on the outside. They don't pass all the way through, and those ones are referred to as peripheral proteins. So what's the function of all these different peripheral proteins and the integral proteins? Well, multiple different roles, but the main one that you do need to know about is involving the integral or transmembrane proteins, because they're involved in the transport. I'll just mention a couple of other things with the membrane. We also have labeled here a whole bunch of sugars. So glyco and carbohydrate, all of these chains that they're showing in green, those are representing sugars or carbohydrates that are either attached to the lipid portion, in which case they're called glycolipids, or they're attached to proteins, in which case they're called glycoproteins. Many, many different roles that are associated with these, and we'll see some of those roles on the next slide here. So I'm sharing this with you, but for the most part, the one that you need to know about for this course is this one. Transmembrane proteins that are involved in transport. So in some cases, they're kind of channels. We'll encounter those. And these ones here can either be gates or carriers or pumps. But there are some other roles. Enzymes allow chemical reactions to take place. So some of these membrane proteins are involved in catalyzing reactions in the cell. Some of them are involved in transmitting messages to the inside or the outside. That's called signal transduction. Cells need to recognize each other, so some of them are involved in recognition. Holding cells together. Most of the cells in your body, they are fixed in place. So joining of cells together will be the roles of some of those. And we saw that both the interior and the exterior of the cell, it's kind of complicated with the extracellular matrix and the cytoskeleton. So a bunch of the proteins will have a role in kind of anchoring the cell to those portions on the inside and the outside of the cell. So back to the phospholipids. The phospholipids made up of two general portions. This prefix here, hydro, just is referring to water. And philic does mean water loving. So there is a portion of the phospholipids that is water loving, and that of course is going to be the portion that is exposed to the water. You have other portions of the phospholipids that are hydrophobic. Phobic means hating, so hydrophobic is then the water hating portion. So we talk about the phospholipids as having the hydrophilic heads that is shown in the picture here. So those are the kind of grayish ovals. And then the yellowish color on the diagram, those are the water-hating or hydrophobic tails that we do see spanning all across in between the two layers. So they've zoomed in on the right-hand side here, so we can see that this here would be outside of the cell, and it is mostly water. Down here, this is inside of the cell, and it is mostly water. So that's why we have all of these water-loving hydrophilic portions that we see here and here. They're exposed to the water. They like that. All of the interior, though, it's made up of these tails. And we can see that each one of the hydrophilic heads is attached to two different tails that make up the phospholipid bilayer. So in between, there is no water, And that is why these molecules arrange themselves. They are what are called self-assembling molecules. Take a bunch of these individual phospholipids, throw them into water, and they organize themselves into a phospholipid bilayer with the tails toward the interior and the heads exposed to the water on the surface. So what about the membrane proteins? Well, they're stuck in, they're embedded in, creating this mosaic-like pattern. The membrane is not a rigid structure, as we will see. It is very, very fluid. It has to be very fluid. Things need to be able to move around. So the phospholipids move around, and the proteins also move around. So the components of the membrane, whether it's the phospholipids or whether it is the proteins, they're free to slide past each other within their layer, but typically they don't flip back and forth between the sides. So this picture that it's showing here, it's saying, well, we have two different cells. We're actually taking two different cells that are both made up of phospholipid bilayers, but they're completely different in terms of their membrane proteins. So the one on the left is a mouse cell, the one on the right is a human cell, and you can actually fuse them together because they're both made up of the same building blocks, which are phospholipids and proteins. But when you do fuse them together, what this shows is that these mouse proteins, they are free to move around anywhere they like, and the same thing with the human proteins. And that shows us the fluid nature of this membrane. So it is referred to as a fluid mosaic model, the structure of the cell membrane. I do have a video if you want to stop and go ahead and take a look at this. The URL that we see here, you can take a look at that, and it sort of shows you the fluid nature of the membrane and how things are able to move around. Here again, we just strictly have the phospholipid bilayer. We don't have any of the proteins at all. So what we mean by the movement is that we can have any of these within this layer that are free to shift back and forth, slide past each other. That's okay. It happens very, very rapidly. This number here is 10 million times per second. What doesn't happen is kind of flipping from the inside to the outside. That very, very rarely does happen. The membranes do need to be fluid. What actually keeps them fluid? Well, the fact that we do have these phospholipid tails, but the lipid tails, they can have different properties. So if we take a look at the one on the right-hand side, first of all, we can see that all of these tails here, they're pretty straight. And those tails allow for these phospholipids to be fairly tightly packed together, and that makes the membrane less fluid. You don't need to know all the details, but these lipids that make up these tails that are kind of straight, they are referred to as saturated hydrocarbon tails in those lipids, in those lipid portions of the phospholipid bilayer. So for these ones here, because they're tightly packed together, it's a little bit more rigid. So these ones here are a little thicker. They don't move quite as freely as the ones on the right or the left-hand side. So the ones on the left-hand side, it says that this membrane is much more fluid. So now if we take a look at some of these tails, they have these little bit of a kink in them here as well. And that's because now the lipid portion of the phospholipid, it is made up of unsaturated fatty acids or an unsaturated hydrocarbon chain. So because we have more space in the membrane when we have all these little kinks here, that's what makes the membrane more fluid. So in a very, very cold environment, these can actually freeze. So we can go from the liquid nature of a membrane to a more solid nature. So to prevent that from happening in very, very cold environments, quite often the cells, they have more unsaturated lipids within the phospholipid bilayer. Something that I didn't mention earlier that's also embedded in the phospholipid bilayer are molecules of cholesterol. So these also interrupt the phospholipid bilayer. If it's a very hot environment, then the membrane might be more fluid. So in a very hot environment, then the cholesterol actually makes it a little bit more viscous. And in a very cold environment, the cholesterol does the opposite. It makes it a little bit more fluid. So those are all of the basic sort of building blocks that we now have. Now let's talk about what goes on in terms of movement across the cell membrane. A couple of terms here. First of all, terms that you should already know. A solvent is a substance that something else is dissolved in. So a solvent is what is doing the dissolving, and it's what a solute is mixed into and dissolves in. The solute is what is being dissolved. The solvent is typically the larger, well, it is the larger, and the solute is the smaller volume-wise is what we're talking about. So an example here, if we take salt and we mix it in water, then the salt is what is being mixed in the water. So the solute, less of the solute, it's what's being dissolved. It is the solute. And the water is what is doing the dissolving. It is the larger of the two components. It is the solvent, and then we end up with a sodium chloride or a saltwater solution. Concentration, you need to have an understanding of what is meant by concentration. Concentration is the number of solute molecules that are dissolved in the solvent. So we can measure it in, we've seen this before, moles per liter. It can also be measured in grams per liter. The more you have, so if I have in this circle here a bunch of solute molecules, the more I add, the more concentrated that solution is going to be. The more you have of the solute, the higher the concentration of that solute. Concentration gradient. If I do have a solution and I put in, if it is just the water, and if I put in a whole bunch of salt at this side, then we have more of it on this side and we have less on this side. So what's going to happen is it's going to move because we have a difference. We have a gradient. And that gradient is going to result in it moving from where it is more over to where it is less until it eventually evens out and we have an equal distribution. Right now we have an unequal distribution in this picture, and that's going to be the driving force to have the net movement of these solute molecules going from where there is more to where there is less. So you should realize that they actually do move in both directions, but the net movement overall, we have more going from the left to the right. So now we're going to be talking, of course, about membranes and movement across membranes. This is kind of a small picture that I do have here, but what it's showing is that separating along the middle of this box, we have a membrane. And this membrane is going to be permeable. It's going to allow these yellow spheres to move from one side to the other. And of course, starting off, we have more of them that are on the left, to the left of the membrane, and we have none of them on the right. So these arrows are showing that they're going to start to move. They are going to move in both directions, but we do have more of them that are moving toward the right until eventually they equal out. They are still moving from the left to the right and the right to the left, but there's no net movement, so now they are in equilibrium. And the picture at the bottom, it shows now that we have two different solutes that are both permeable in this membrane. The yellow can go to the left, well, it can actually go in both directions. Yellow can go to the right. The purplish one can go to the left. So that is going to happen. They're going to keep moving around until, again, eventually we have the same number of both of these solutes on either side of the membrane. So what is driving their movement, though, is just simply the difference in the concentration between the two sides. So the first kind of transport that we'll talk about is the kind of transport that doesn't involve any energy, and that is called passive transport. No energy required, no work is being done. This is a little bit deceiving. The energy is in fact just the concentration gradient, where you have more on one side, less on the other. That's what's going to be driving it, but there's no external energy source other than the difference in the concentration. So the first one, simple diffusion, is the movement of molecules from an area of higher concentration to an area of lower concentration across the membrane. So this is movement. We're talking about cells now, movement across the membrane from where you have more to where you have less. So this is our phospholipid bilayer. We have no proteins involved in this phospholipid bilayer. It does not require any kind of a transport protein. We're moving it directly through the membrane from where we have more to where we have less. Now, unfortunately, very, very, very few things can actually do this. In order to move through the membrane, they typically have to be small. They can't have a charge. They need to be hydrophobic so they can go through the interior of the membrane, which is hydrophobic. And quite often what goes along with that is we talk about them as being non-polar. So again, very few things actually meet these criteria. Any ions that we have, like a sodium ion, that's not going to pass through. Any proteins that we have, they're simply too big. They're not going to be able to pass through. So some things that can pass through are some small lipids and some gases. Oxygen can pass through. Carbon dioxide can pass directly through the phospholipid bilayer by simple diffusion. But we need some other processes in order to move other things through the membrane. Things that are larger, things that have a charge, and things that are polar in nature. So now what we have is facilitated diffusion. It's still diffusion. We're still moving from where there is more to where there is less, but now we need a little bit of help, and that's why it's referred to as facilitated. And facilitated diffusion involves transport proteins, which might be channels or they might be carriers. So the picture that we have at the top here, this is showing a channel. So it's still diffusion because these solutes, we have more on the top side, we have less on the bottom side. Only they can't go through the phospholipid bilayer. Why? Well, maybe they're charged, or maybe they're too large, or maybe they're hydrophilic, or maybe they're polar. So now the only way that they can get through is by passing through this channel from one side to the other. Still doesn't require any energy because they are still moving what we call down their concentration gradient or with their concentration gradient from where there is more to where there is less. Same principle with this one here, only now when the solute binds to this protein, it causes a structural change in the shape, and that just allows it to be released on the other side. Still no energy is required, so this one is what's referred to as a carrier or a gate, but still we are moving from where there is more to where there is less, and it does involve a membrane protein. So it is diffusion, but it is facilitated diffusion. Osmosis is referring specifically to water. It is still diffusion, only it is the diffusion of water. It's passing through a selectively permeable membrane is what we are talking about, like the cell membrane or an organelle membrane. So it is still talking about going from where there is more, and here is the water, from where there is a higher water concentration. If there is a higher water concentration, that means there has to be a lower solute concentration. It's an inverse relationship. More water means less solute. So we're moving the water from where there is more to where there is less of that water or more of the solute. So water passes through, not through the phospholipid bilayer. It cannot do that because water is polar. Anything that's polar, like water and glucose, can't pass through the phospholipid bilayer. It has to be facilitated diffusion. In this case, the protein has a name, and it is called aquaporin, but it is facilitated diffusion. So if we take a look at this U-tube that we have here, this is an artificial membrane, but it is a selectively permeable membrane. And this is a membrane that only allows for the movement of water. So all of these purple solutes that we see here, they can't actually pass through the selectively permeable membrane. A couple of terms that we'll come back to here. A hypotonic solution is a solution where you have more water, and because of that, you have less solute. Hypertonic solution is where you have less water and you have more solute. So because we have higher water on the left-hand side than what we have on the right-hand side, the water is going to move through from the left to the right. So if we take a look at the right-hand diagram, this is what happens once we reach equilibrium. So again, these solute molecules haven't been able to move, but the water has. So we actually have the water level on the right-hand side of this U-tube that does in fact raise up, and it is higher because of the diffusion of water, osmosis of water through this selectively permeable membrane. Active transport. So now, for the first time, we're going to be moving things where they actually don't want to go. Instead of going from where there is more to where there is less, active transport is going to be the movement of materials from where there is lower to where there already is more. So they actually don't want to go in that direction. We call this against the concentration gradient or up the concentration gradient, whereas with diffusion, it was down or with the concentration gradient. This does have to involve membrane proteins, and it has to involve energy. Oops, we don't see it in the picture here. It's hidden, but it does involve energy, and that energy is in the form of ATP, which we do see in the picture here. So in this case, this is our hydrogen ion. Hydrogen is small enough, but it has a charge if it's the hydrogen ion, so it cannot pass through the phospholipid bilayer. We can see that we already have more of these hydrogen ions on the right. We have less of them on the left. So we're moving the hydrogen ions in a direction that they don't want to go to. They actually want to go in this direction. That would be diffusion, but of course we know they can't pass through there. But here we want to move them in the opposite direction to where there already are more. In other words, what we want to do is we want to concentrate them. We want to concentrate more of these protons, hydrogen ions on the right-hand side. So we're using this proton pump, and that's exactly what it's going to do, is it's going to move them from the left-hand side to the right-hand side, and that requires energy because they're going against their concentration gradient. And if you do want to take a look at a video here, this is a URL, and it'll take you through some animations that show all of these different transport processes. A little bit more terminology to kind of finish things off here, referring specifically to the movement of water. So a cell can have the ability to either gain or lose water depending upon the environment that it is placed in. So inside of a cell, there will be water, but there will also be solute. So if you have on the outside of the cell water and the same amount of solute, then there's going to be no net movement of water. And that's what we call an isotonic solution. Yes, there will be some water that's moving in, but there also will be some water that's moving out, and it's going to be equal. There is no net movement of the water. If we do have more solute outside of the cell, then that's going to cause the water to move out. So if we have a hypertonic solution, hyper does mean high, and it's referring to the solute. So solute concentration is greater than inside the cell. Inside the cell less, outside the cell more. So it's not the solute that's going to be lost, it's the water. So water is going to move out of the cell. Hypo is if you have the opposite. So now this is a low solute concentration, more water. So in this case, the cell is going to gain water. Let's take a look at a couple of pictures here. These are animal cells at the top. In fact, these are your red blood cells. So if we take one of your red blood cells, or a number of your red blood cells, and put it in pure distilled water, I'll put it as DO2. In this case, there's zero solute outside of the cell if it is distilled water, and there is solute inside of the cell. So water is going to try and dilute that. It's going to move into your cell, it's going to swell up, and it's going to burst, and that is what we call cell lysis. If you have an equal solute concentration inside and outside of the cell, and an equal water concentration inside and outside of the cell, then it's going to remain in this normal shape. If you do put a cell in a hypertonic solution, then water is going to leave and it's going to shrivel up. It's somewhat similar in plants, only plants, of course, have a cell wall around the outside, and that's going to prevent cell wall. That's going to prevent the cell from swelling up and bursting. So if we do put the water in a hypotonic solution, all of these in this first column here, this is in a hypotonic solution. Water will move into the cells. This one won't burst because it has a cell wall, and in fact, this is normal. It keeps the plant upright and rigid. If you do have isotonic, then the plant cell actually starts to wilt, which is called flaccid. And if we have a hypertonic solution, which is essentially putting the plant in a salt solution, the water moves out, the cell will shrivel up, it's called plasmolysis, and that plant cell will eventually end up dying because of that.